Immune system. Almost everything around us teems with pathogens or agents that cause disease, from the air, the water, the foods we eat, and on the things we touch. Nevertheless, most of us remain healthy because of our immune system, which is our body's system of defenses to fend off microbes that exist to harm our health. The body's first defense against microbes are the skin and the mucous membranes of the digestive, respiratory, reproductive, and urinary systems. The dry, dead cells filled with hard protein that cover the outer surface of the skin prevent most microbes landing on the skin to enter our body. Mucous membranes are protected with mucus, which contain antibacterial enzymes which destroy bacteria and physically trap microbes that enter the body. Cilia sweep up the mucus until it is dislodged from our body. Microbes that breach our external barriers run into our innate immune cells, white blood cells. Standing parties of phagocytic and natural killers travel through the interstitial fluid in the capillaries to where the bacteria have gained entry, ooze out through these narrow openings, then macrophages, large phagocytic cells, ingest bacteria and other particles. Neutrophils, the most abundant white blood cells, like macrophages, also engulf foreign invaders such as bacteria and foreign fungi. Neutrophils secrete molecules that attract defensive cells to the invaded site. Natural killers, on the other hand, identify and destroy body cells that had been infected by viruses or had become cancerous. There are also proteins that attack or impede production of microbes, synthesized like interferons by virus-infected cells diffused to neighboring healthy cells, thus stimulating them to produce other proteins that inhibit viral infection. Another part of our innate immunity is the complement system, a group of proteins that can act together with other defense mechanisms. Its main function is to trigger inflammation and to kill microbes by a cascade of steps that may lead to the lysis or bursting of the invaders. Damage to our body tissues elicit a reaction called the inflammatory response. Whenever our skin is damaged, it becomes red, swollen, and warm. That reaction is inflammation. Damaged cells release chemical alarm signal histamine into the wounded area, making these capillary walls leaky, causing the fluid from the capillaries into the tissues around the wound and for it to become inflamed. Meanwhile, other chemicals released by injured cells initiate blood clotting. Blood clotting blocks damaged vessels, preventing microbes from escaping into the bloodstream and seals the wound from the outside to limit the entry of more microbes. Still, other chemicals, some released by wounded cells and others produced by microbial invaders, attack macrophages, neutrophils, and other phagocytic cells to the wounds where they engulf microbes, dirt, and tissue debris. The pus that accumulates largely consists of microbes, tissue debris, and white blood cells. If enough microbes survive the inflammatory response and infect larger areas of the body, a fever may be triggered. While severe fevers are dangerous, even fatal, average people of 48 degrees Celsius, 100 to 102 degrees Fahrenheit are beneficial. It is a systemic inflammation that increases white blood cells to stimulate phagocytosis and hasten tissue repair. The lymphatic system, both involved in innate and adaptive immunity, consists of branching networks of vessels, numerous lymph nodes, the bone marrow, and several organs. The immune system consists of about 2 trillion of specialized white blood cells called lymphocytes. Many are distributed throughout the body in the blood and lymph. Others are clustered in specific organs, particularly in the thymus, lymph nodes, and spleen. The immune response results from the interaction of several types of lymphocytes and the molecules they produce. The lymphatic system has two main functions: to return tissue fluid back to the circulatory and to fight infection inside our body. Infection, once in the circulatory system, is carried into the lymphatic vessels, as lymph then delivers it to locations where it can be cleansed of debris and checked for the presence of pathogenic organisms. Lymphatic vessels depend mainly on the movement of skeletal muscles to squeeze the lymph along our body. There are also these one-way valves that prevents the backflow of the lymph. There are also lymph nodes, little round organs packed with lymphocytes. They are testing stations, monitoring the samples of blood plasma, and produce lymphocytes, sending them to the bloodstream to destroy the invaders when they filter the lymph so that they only return clean fluid back to the circulatory system. More than 2,000 years ago, the Greek historian Thucydides recognized that occasionally some people contract a disease, recover, and never catch that particular disease again. Thus, they become immune. Immunity to one disease, however, doesn't mean immunity to another. Thus, the immune system attacks one type of microbe, overcomes it, and provides future protection against that same microbe, but no other microbe. This is why it is called a specific defense against invasion. Antibodies are proteins found in blood plasma that attaches to one particular kind of antigen, a foreign protein. Vaccination is a way to trigger a body's immune response without becoming sick. The vaccine contains weak or dead organisms that usually do not cause illness. The body is able to destroy the pathogen in the vaccine and produce memory cells when the body develops immunity to a specific illness. Thus, immunization has occurred. If the body is ever exposed to the same pathogen, the memory cells react quickly and efficiently before the symptoms appear. The immune response results from the interaction of several types of lymphocytes and the molecules they produce. The immune response is provided chiefly by two types of lymphocytes, B cells and T cells. Like all white blood cells, B cells and T cells arise from cells in the bone marrow. Newly forming T cells migrate to the thymus, hence the name T cells, and differentiate. B cells, on the other hand, differentiate on the bone marrow cells, hence the name B cells, and later migrate typically either to the spleen or lymph nodes. Immune responses produced by both B cells and T cells consist of three steps: recognizing invaders, blocking an attack, and retaining a memory of an invader to ward for future invaders. B cells and T cells together provide a dual defense. The first type, the humoral immune response, defends primarily against bacteria and viruses present in body fluids. This response can be passively transferred by injecting antibody-containing blood plasma from an immune individual into a non-immune individual. The second type, cell-mediated immune response, is where T cells attack infected cells while other T cells function by promoting phagocytosis and stimulating B cells to produce antibodies. In both cells, there is recall that antibodies and antigens are both proteins. Most antigens are proteins or large polysaccharides that protrude from the surface of viruses of foreign cells. These antigenic molecules has these antigenic terminants, also known as epitope, are these small surface exposed region of an antigen that an antibody usually recognizes and binds to. These terminals are complementary to a specific antibody's antigen binding site. The immune system's ability to defend against a wide variety of antigens depends on a process known as clonal selection. When an antigen enters the body for the first time, it will be shipped off to the lymph nodes, and there, its antigenic determinants on its surface binds to the few B cells that happen to have complementary receptors. Other B cells without the appropriate binding site will be unaffected. The selected B cells will then be activated to grow, divide, and differentiate to form a new group of effector cells through mitosis. These effector cells are plasma cells that secrete antibody molecules that will circulate in the blood and lymphatic fluid but would only last for some 4 to 5 days. After cells become memory cells that had undergone a series of phenotypic changes, making them more efficient on attacking the same antigen if it is ever presented again in the future, meaning there is always a ready supply of cells with appropriate specificity to fight infection. These memory cells can live for many years, and this increased efficiency of memory cells to fight infection underlies the enhanced secondary immune response. Memory cells confer lasting immunity against a pathogen, which is the basic principle behind vaccination. Antibodies are composed of peptide chains, one large heavy chain and one small light chain on each side of the Y. Both heavy and light chains consist of constant region similar in all antibodies of the same class and a variable region that differs among antibodies. The combination of light and heavy chains result to an antibody with two functional parts, the arms and the stem of the Y. The variable regions of the arms form highly specific binding sites for large molecules. Each binding site has a particular shape and electrical charge, so only a few or perhaps only one type of molecule can fit in and bind to the site in general. Antibodies do not kill pathogens. They only mark antigens by combining with it to form an antigen-antibody complex, which would make it easier for other components to kill the antigen. There are several mechanisms to destroy an invader. The first one is neutralization, where antibodies bind to surface proteins on a pathogen, thereby blocking its ability to infect host cell. The second one is agglutination, where antibodies clump invading cells together. The third is precipitation, where antibodies link dissolved antigen molecules together for antigen to precipitate or separate in solid form from the surrounding liquid. These first three make it easier for phagocytes to capture the invaders. There is a fourth, however, activation of the complement system or opsonization, where proteins attach to a foreign cell, then forms a complex that pokes a hole in the plasma membrane of the cell, causing it to lyse. An antigen, such as a protein, has several different sites that can react with different antibodies. Monoclonal antibodies are antibodies that react with only one particular site on an antigen called epitope. Consequently, they are highly specific and have applications in both diagnostic biology and cancer therapy. The trick to making monoclonal antibodies is the fusion of two cells to form a hybrid cell. Two common uses of monoclonal antibodies are in home pregnancy tests and in the diagnosis of diseases. Macrophages lead the cell's mediated immune response by engulfing and digesting foreign invaders. Combining peptide fragments from the invaders with some of its proteins, the macrophage builds a patchwork of antigen complexes on its surface. When smaller white blood cells called helper T cells encounter the macrophage, those with matching receptors bind to its surface. These helpers then multiply and secrete chemicals, which called different cells into battle, especially the cytotoxic or the killer T cells. The macrophage or an infected cell activates only those cytotoxic cells with receptors that match the antigen complexes stimulated by chemicals from the helper T cells, rapidly divide into an army of clones, all designed to fit the specific invader that trigger the response. When killer cells dock with an infected cell, they bombard it with lethal toxins, then move on in search of other toxins. Immunodeficiency disease results from the inability of the immune system to mount an effective immune response to infection. Probably the most common and most devastating immune deficiency disease acquired is immunodeficiency syndrome or AIDS. Two viruses named human immunodeficiency viruses 1 and 2, HIV-1 and HIV-2, cause AIDS by infecting and destroying helper T cells. Though AIDS patients produce HIV antibodies, for unknown reasons, they do little to prevent progress of the infection. One hypothesis is that HIV spends so much of its life cycle inside the cells that it is the cell that is exposed to the antibodies. AIDS does not directly kill, but as the helper T cell population declines, the AIDS patient becomes increasingly susceptible to other diseases. HIV-1 and HIV-2 are retroviruses, viruses that have RNA, not DNA, as their genetic material. HIV consists of an outer envelope that taken from an infected cell's plasma membrane as the virus leaves the cell and two protein capsules, the innermost in which it contains RNA and an enzyme called reverse transcriptase. The outer envelope binds to the plasma membrane of the helper T cell and allows viruses to invade the cell. Once inside, the reverse transcriptase copies the virus's genome over into the DNA because the normal direction of transcription in living cells is DNA to RNA. The virus direction RNA to DNA is dubbed reverse transcription. The infected cells all metabolic machinery then uses the viral DNA copy to make more viruses and more reverse transcriptase. This multiplication of HIV viruses eventually kills the infected helper T cells. HIV mutates at a high rate during replication, making it very hard to cure. Current HIV treatment only prolongs life of an HIV positive person, but it is temporary. The lymphocytes has the ability to recognize the body's own molecules to distinguish self from non-self, thus enabling our adaptive immune response to battle foreign invaders without harming healthy body cells. The key is that each person's cells have a unique collection of self-proteins on the surface, seemingly molecular fingerprints that can be recognized by the immune system. Because of this, the immune system can also distinguish our own cells from other people's. Genes at multiple chromosomal loci code for major histocompatibility complex molecules, the main self-proteins. This ensures that no two people can have completely matching sets of MHC self-proteins. Autoimmune diseases is a disease where the body actually attacks itself. Remember that our bodies have a defensive system which normally guards us against invaders, but in this case, the body actually loses the ability to differentiate friend from foe. The tenacity of our immune system actually turns against our own organs. The ultimate result may lead to a devastating loss of an organ's function, which could lead to a person's death. The most common types of autoimmune diseases are type 1 diabetes, rheumatic heart disease, rheumatoid arthritis, multiple sclerosis, lupus, and Parkinson's disease. There are many aspects of autoimmune disorders that are unknown at this time, but one thing is certain. It is that one of the basic mechanisms behind the self-destructive behavior of the body is a process called molecular mimicry, from the word mimicry meaning to mimic. It is what happens when some of the foreign cells look so much like our own that our immune system mistakenly assumes that our own cells are foreign. The body becomes confused and can no longer tell the difference between the invaders and its own cells, thus attacking everything, even our own cells. For a person with allergies, the immune system takes harmless substances known as allergens as a potential threat. The first time the immune system encounters an allergen, it reacts by activating special cells. These cells make large amount of proteins called antibodies that is specific to the type of allergen. These antibodies then attach to other immune cells, making them sensitive to specific allergens. The next time the allergen enters the body, these sensitive immune cells quickly attack. They attach to the allergen, which causes the release of natural chemicals, including histamine. These chemicals enter the surrounding tissue and cause the early symptoms of allergy.